You will need to read pages 199 to 203 on your own. You'll need to know the terms in bold. You should be familiar with the definition and pretty much how to use them. There are a lot of terms. You should know stuff like genes are the segmented DNA in specific locations on specific chromosomes that code for specific proteins. Yeah, we're kind of going to talk about how you get to those genes, how you use those genes. Remember homologous chromosomes and alleles and meiosis from several other chapters. We'll also need to know about a monohybrid cross involving one gene for one trait. There's usually two alleles, but it's only one gene. A dihybrid involves two genes for two different traits. Usually there's two alleles for each of the two genes, so it looks like we're crossing four things, sort of. A phenotype is a measurable characteristic, observable features, tall plants, short plants, purple flowers, white flowers, right-handed versus left-handed, things you can see. The genotype is the actual genetic makeup or formula for a particular phenotype. It's going to be represented with letters. We use the letters of the dominant gene. So if tall is dominant over short, we'll use T's for tall, big T's for tall, and little t's for short. Homozygous is having the same alleles. So if you've got a tall allele and a tall allele, you'd be homozygous. If you've got a short allele and a short allele, you'd be homozygous. Heterozygous has different alleles. If you've got a tall allele and a short allele, you would be heterozygous. How big are you? Yeah, we'll talk about that eventually. A dominant gene is one that will always produce its phenotype. If you've got at least one big letter in the genotype, you'll have the dominant phenotype. Big T is going to stand for tall. If you've got even one big T in your genotype, you would have a dominant phenotype and you would be tall. So my example with the heterozygous tall allele and the short allele, because you've got at least one dominant, you would be tall. If you were a plant anyway. The recessive is the one that produces its phenotype only in the homozygous genotype. If you're going to be short, that's the recessive. It can be kind of hidden by the dominant big T. So in order to get a recessive phenotype, you have to have two little letters. Little t, little t. Pages 202 and 203 had a few questions in them. For example, on page 202, they ask how many genes are responsible for type O positive blood. That would be two different genes. There's a gene for the type A, type B, type O, type AB, the basic blood type, and then there's a second gene that controls whether you're Rh positive or Rh negative. So it's actually two different genes. In the letter D section, tall plants are dominant over short pea plants. What letter would you use to represent the short genotype? Sometimes students guess little s's because it is short, but really we're going to be using a little t. What? Tall pea plants are dominant, so we're going to use t's for tall and short. We'd use a capital T for the tall dominant phenotype or genotype, and the small little t for the short genotype. Yeah, it's kind of confusing, but when you're doing a lot of different genes in one problem, it's easier if you can tell which pair goes together. Tall versus short, you can tell because they've both got T's in them. On page 203, letter H, there's what is the phenotype of the heterozygous big T little t pea plant? That would be tall. I think I answered that one already, but just in case you missed it, big T little t is heterozygous and would be tall. What's the genotype of a short pea plant? Well, you would use little t's in order to get the short recessive phenotype. In order to show up the phenotype, you'd have to have the little t genotype. On page 204, the genetics problems actually start with letter A. An example, consider the dominant allele big T for tall and the recessive little t for short. We're supposed to cross a homozygous tall plant with a homozygous short plant, determine the phenotypes and genotypes and weird stuff. Well, before we get too confusing, the basic thing you want to do is start thinking about your letters that you want to use. The question did say that tall is going to be dominant, so we should use a big T for tall, which means we should use a little t for short. The next thing we want to do is think about what the parental genotypes are. It said we were crossing a homozygous tall plant to a homozygous short plant. What are the genotypes that we're crossing? You think about it for just a second and I'll go on. Hopefully you figured out that the genotypes are big T, and little t, little t. Homozygous means they're the same. Tall, we said we were using capital T's, so we should use capital T for homozygous tall. Homozygous short, we were using little t's for short, so homozygous short would be little t, little t. Keep in mind, sometimes they don't always say homozygous recessive. Sometimes they just talk about recessive genotypes. Kind of like your mom may just call you by your first name, unless she's mad at you and then calls you by your full name. The full name would be homozygous recessive. Most of the time we just call them recessive. In order to see the recessive phenotype, you have to have the homozygous little t in this case, genotype. The next step we need to do for genetics problems is to figure out the gametes for each parent. That big T homozygous parent, you could do meiosis and duplicate the chromosomes and split the homologous chromosomes into different cells and then split the sister chromatids into different cells, but you know what? If you've got big T to start with, you can only give a big T or a big T. Same thing with the little t genotype. You could duplicate everything, you could split it all apart, but it doesn't matter how you slice it. It's going to give a little t or a little t. The cheating way for the monohybrid crosses, take the genotype and put a comma between the letters. We had big T, we now have big T, comma, big T. We had little t, the gametes would be little t, comma, little t. Yeah, it really is that simple. It kind of cheats, but there you go. Put a comma in between the two. If you're in my class and doing dihybrid questions in lecture, that does not work. You can do a Punnett Square from question A. You would put the gametes from the first parent on the top and the gametes from the second parent down the side. Kind of look on the left-hand square. We've got the big T on the top. We got the little t on the side. What you officially do is bring the big T's down and the little t's across, or kind of meet in the middle. So if you look at the Punnett Square on the right, we've got the big T's on the top, got the little t's on the side, and all of them end with a big T and a little t in the box. When you combine the gametes, the first parent can only give a big T and the second parent can only give a little t, so you didn't really have to do a Punnett Square, although I was just showing you how to do it for practice here. The questions go on and says the resulting F1 genotypes can only be big T, little t. Just what we got in the box. But what's the phenotype of an individual that is big T, little t? Is it tall or short? It's got at least one big T, it is going to be tall. Tall goes in the blank. Sometimes they ask about counting the phenotypic and the genotypic ratios. The phenotypic ratio is how many of the different phenotypes you've got, and the genotypic ratio is how many of the different genotypes have you got. On this problem, it's not too bad because they're all one thing. The phenotypic ratio is all tall, and the genotypic ratio, where you're counting the letters, is all big T, little t. However, the question actually continues on letter B. See, it turns out that plants can self-fertilize, where the male part of a flower can fertilize the female part of the same flower. Huh? What? In genetic terms, self-fertilize means cross it to the same genotype. That's just kind of one of those code words out there. So when question B says to allow the previous F1 generation to self-fertilize, what are the genotypes we were crossing? Well, remember the definition for the F1 is the kids, the things in the boxes on the previous slide, was a big T, little t. We're going to let that self-fertilize, so we're going to cross it to another big T, little t. Like I said a second ago, the parents of this cross is big T, little t, cross 2, big T, little t. Don't forget there are some blanks on page 204 to write this in. The next question is, what are the gametes? You could do meiosis, or you could cheat and put a comma in between the big T and the little t. And then since the other parental genotype is big T, comma, little t. Both of the gametes are the same thing there. Ultimately, in the lab book, the gametes from the first parent have been put along the top of the Punnett Square in figure 20.5, and the gametes from the second parent are down the left side, and we'll be working with that Punnett Square in just a second. The big T, little t from the first parent have been put along the top of the Punnett Square box, and the big T, little t have been put down the side of the Punnett Square box. Why the Punnett Square box is in 3 different colors, that I'll never know. But what you need to do is bring your big T at the top down and your big T from the side over so that your upper left-hand box has a big T, big T. In the kind of second box, you would bring the little t down, you would bring the big T over. By convention, we usually put the big T first. That's fine, we can usually read through it either way. But the upper right-hand box has a big T and a little t in it. For the third box, bring the big T down, bring the little t across, and you end up with a big T, little t, heterozygous. And then in the last box, you bring the little t down and the little t over, and you end up with a little t, little t. You'll also want to think about what the phenotypes are. Is the first box tall or short? Is the second box tall or short? What about the third one? What about the fourth one? You should fill those in on your lab book. Hopefully, you figured out the phenotypes. Big T is tall. Big T is tall. Big T is tall. And little t is short. Underneath figure 20.5, there are some more questions. What's the phenotypic ratio and what's the genotypic ratio? The phenotypic ratio is when you count the phenotypes. You'd be counting how many of them are tall and how many of them are short. There's 1, 2, 3 of them that are tall and 1 of them that's short. We usually just call that a 3-to-1 ratio. The genotypic ratio counts the different genotypes. Notice that homozygous dominant, big T, is different from heterozygous dominant, big T, although they both end up being tall. Now, little t is obviously a little different because he ends up with a short phenotype, but you would count how many are big T. 1. How many of them are big T? 2. And how many are little t? Just 1. We'd call that a 1-to-2-to-1 ratio. Usually geneticists will put it in order with the 1 big T, the 2 big T's, and the 1 little t. Sometimes students kind of put it in numerical order of 2-to-1-to-1. You might want to make sure you put your letters after it so that everybody can tell which one you are really talking about. At the bottom of page 204, letter C is our kind of third problem here. In guinea pigs, black fur is dominant over white fur. A black male guinea pig is mated to a black female guinea pig. Over the course of 15 months, they have 27 black offspring and 9 white offspring. The offspring illustrate what phenotypic ratio? Ooh, this sounds complicated. Yes, it kind of is. We'll work through it. It's not that bad of a deal. First of all, let's establish our coats. The question did say that black fur is dominant over white fur, so what letter are we going to use for black fur? Let's try a big B. It's dominant and black. What letter are we going to use for the white fur? Be careful, you still use a little b to show that white fur is the opposite of the black fur. Now, it did say there were 27 offspring with black fur and 9 offspring with white fur. Math class, reduce down to its lowest common denominator. Divide both sides by 9. 27 divided by 9 is 3. 9 divided by 9 is 1. This is going to be a 3-to-1 ratio. So, what what were we crossing? Yeah, we'll get there. But when it did ask about the offspring illustrate what phenotypic ratio, it was a 3-to-1 ratio, even if there wasn't a blank to write it down in. The first real question with the blank is, what's the genotype of the white offspring? Remember, the white fur is recessive. In order to see white fur, you'd have to have a little b, little b. We can figure that one out too, without too much trouble. The genotype of the white offspring is little b, little b. Where must each little b have come from? Each parent? Each of the parents would have to have a little b on the mommy guinea pig and a little b on the daddy guinea pig so that they come together to make the little b on the little baby guinea pig. But each parent is black, so what are each of the parents' genotypes? How about big B, little b? On page 205, the lab book does ask what are the genotypes of the parent. They do have to have a little b. Each of them has to have a little b so that the offspring can have that white fur. But the question did say both of the parents were black, so they're going to have a big B as well. Both of the parents are going to be big B, little b, crossed to big B, little b. Once you make the Punnett Square, it's a little easier to see that the genotypes of the black offspring are going to be big B, and big B, little b. So, kind of going forward on the question, the P1 or parental generation, we did say each of the parents was going to be a big B, little b, crossed to big B, little b. The gametes, we were just putting a comma in between big B, comma, little b, and big B, comma, little b. There's the Punnett Square. There's a spot to write it down. I've already got it drawn out here. Underneath, they ask again, what's the genotype of the white offspring? Was little b, little b. What are the genotypes of the black offspring? Was big B, and big B, little b. The offspring illustrate what phenotypic ratio? Is 3-to-1 ratio. There's 3 of them that are black and 1 of them that would be white. Partially the reason we did that problem is to show you another question. If you cross 2 dominant phenotypes and you get dominant and recessive in the offspring, what cross did you make? It's really the same question, but there's usually on a multiple choice exam, like our lab exam, we could ask you if you crossed 2 black-furred guinea pigs and you got some dominance and some recessive in the offspring, what cross did you make? And a lot of people will spaz on the question. Don't forget it is a multiple choice exam, so you would have all sorts of different choices, say A, B, C, D, and E. If you don't see immediately that it's crossing 2 heterozygous individuals, you could work out all the Punnett squares from all the different answers. For example, letter A is crossing big B to big B. You get a lot of offspring with a bunch of big B's. What color is big B? Black. And big B is black. And big B, I can't even talk. You get a whole bunch of offspring with black fur. Letter B was crossing a homozygous dominant to a heterozygous, and you still get a bunch of black offspring. Either they're homozygous black or they're heterozygous black. Those aren't it. Letter C was crossing 2 heterozygous individuals, and you do get some black offspring, black. Oh, hey, there's a little b, there's a short offspring. Letter D, you cross a heterozygous with a recessive. Now, in this case, you do get about half dominant, half recessive. Remember our original question, you did get a 3-to-1 ratio. But there's actually another problem with letter D. What's wrong with letter D? What phenotype is big B, little b? Black. And you were crossing it to a little b. What phenotype is that? White. Question said we were crossing 2 dominants. There's a problem with letter D. We didn't cross 2 dominants. What's the problem with letter E? We crossed 2 recessives. Yeah, well, it also doesn't give dominant recessive in the offspring. And yes, letter E, if you cross a whole bunch of little b's, you get a whole bunch of little white guinea pigs. But the trick with D is you crossed a black parent with a white parent, and you got half black offspring and half white offspring, which was not quite what we were talking about in the question in the lab book. But if you didn't immediately see that letter C was the correct answer, you can always work through the problems. You can always work through them and see what the answers are. Oops, I kind of gave away the answer on this slide. What's wrong with D and E? The fact that you are not crossing two dominant phenotypes. But honestly, the only way you can cross two dominants and get some dominant and some recessive in the offspring is letter C, to cross the two heterozygous individuals. You can work through all sorts of combinations, but the only one where you cross two dominants and get dominant and recessive in the offspring is to cross two heterozygous individuals. Think about it, because it's a popular question on quizzes and lab exams. Two dominants, you get a dominant and recessive in the offspring, it's got to be crossing two heterozygous individuals. The next question on page 205, letter D, says a test cross crosses a homozygous recessive parent to a parent of an unknown genotype. Thus, the unknown genotype can be determined. We've got some codes here again. If you're talking about a test cross, that basically means cross a dominant to a recessive. The test in the test cross is the recessive. So what we're going to do is we're going to test cross a tall pea plant. What's the phenotype of the other plant? That's going to be that test cross. That's going to be the recessive. The test cross parent is always the homozygous recessive, in this case a short pea plant. So what's the genotype of the short pea plant? Ooh, better get our codes ready. If we were saying that tall is dominant, we're going to use big T's for tall, so we'll use little t's for short. So our genotype of the short pea plant is going to be little t, little t. Is the unknown genotype going to be dominant or recessive? Well, we just figured out the genotype of the recessive. Maybe we should guess dominant. Yes, we should have guessed the unknown genotype is dominant. That's because a dominant, remember, could be homozygous dominant, big T, or it could be heterozygous dominant, big T, little t. There's the question in the lab book. The answer is dominant, and why? Because it could be homozygous, or it could be heterozygous. It could be big T, or it could be big T, little t. We don't know. We're going to have to test it and find out. Oh yeah, that's why they call it a test cross. We're going to cross it to a recessive and test and see what the dominant really is. So back to the question in the lab book, the P or P1 parental generation is a T with a dash. What the heck does that mean? The T with the dash, that dash just means we don't know if it's big T or big T, little t. So we're going to put a big T and a dash because we don't know what it is. I still think we should have put a big T and a question mark. That would make it a little more obvious that we don't know what the second letter is. But we're going to cross a big T and a dash cross to a little t, and we're looking to see if all the offspring are tall. If the F1 offspring are all tall, then the genotype of the tall parent was, I don't know. Well, we're going to have to do two tests on it. We can cross big T to little t, and then we can do big T to little t, and see which one gives all tall offspring. Do your Punnett squares if you need to. I'll wait for just a second. And no, there's really only one Punnett square box on page 205, but there's plenty of room to write the other one if you needed to. If we were crossing big T to little t, there's the Punnett square on the left, and you can see there's a big T, little t. Hey, that's tall. The next one's tall. The next one, hey, those are all tall. Hey, I bet it's the first cross. Well, just in case, let's do the other one, where you cross the big T to the little t, in which case the upper left and the lower left boxes do have some tall offspring. Great. Uh-oh, the boxes on the right-hand side are going to have some short offspring. That's not it. That Punnett square on the right-hand side did not give what we needed to. So the one that gives all tall offspring is going to be big T, crossed to little t, our test cross, and we do get a whole bunch of heterozygous but tall offspring. Be sure to flip the page, because on page 206, the question is now, but if the F1 offspring are all tall and short, what's the genotype of the tall parent then? Well, it's not that square on the left, because that gave all tall offspring. But what if we cross the heterozygous big T to the little t? Again, the boxes on the right-hand Punnett Square, but the left-hand side of that one, does give all tall offspring. And on the right-hand side, it gives some short offspring. So we've got tall and short offspring. So if we're doing the cross on page 206, now we've got a big T. Because you're crossing it to a recessive, it kind of depends what the offspring end up. It kind of depends on what alleles the original parent had. If the parent had all tall alleles, big T, then all the kids end up being tall. If the original parent had big T, kind of half the alleles were big T and tall and half the alleles were little t and short, then you do end up with half the kids being tall and half the kids being short. So you can test things. Now, honestly, we do that a lot in plants and some species of animals, but for next week's lab, when we start talking about some human genetics, we don't actually do a test cross. We may look back at somebody who accidentally did a test cross, but we don't start going, all right, you and you and you and you, go ahead and have some kids and see what happened. No, we don't do that. But we might look back at an accidental test cross that it's already happened and see if we can determine some genotypes there. On page 206, number 4, probability. One of the most horrible, awful, nasty parts of genetics problems is that they allow you to predict the chances of a particular phenotype occurring. Wait, that wasn't quite what it said. Well, a lot of students find it to be one of the most horrible, awful, nasty things there. But genetics is really a matter of probability. I think that's why so many students don't like genetics, is it's math class. And yeah, it's some of the harder math class we'll do. I'm sorry about that. As a simple example, consider the probability of coming up heads with a single toss of a coin is going to be 1 chance in 2 or 1/2. When we talk about probability, that's really how many of whatever the question asks for divided by the total. So in the lab book where they're talking about apply this to a question of probability of a certain genotype occurring, for example, in a cross between 2 heterozygous tall plants, big T crossed by big T, there's a spot for the Punnett Square. Hopefully you can do that one. Maybe you need to stop the video for just a second. The book does say in the cross between the 2 heterozygous tall plants, the genotype of big T occurs in 1 out of the 4 boxes. So the probability of big T is 1, big T, out of the 4 boxes would be 1 out of 4. Yes, I know that's 25%, but we're going to get into some weird percentages at some point, and I don't have a calculator handy. So deal with it, we're staying with fractions. Anyway, the question then asks what's the probability of an individual from that problem having a genotype of big T. There are 2 big T offspring in the boxes out of 4 boxes. 2 out of 4 would be 1/2, would be 50%. What's the probability of being little t? P.S. If you have the green book, it's got a typo in it and it says big T. It should be little t. What's the probability of being little t? There's 1 little t out of 4 boxes would be 1 out of 4, or 25%. So there's probability there. Now to extend this idea, consider the probability of flipping heads twice in a row with our imaginary coin. The chance of flipping heads the first time is 1/2. The same is true for the second flip. The chance or probability that we flip heads twice in a row is not 1/2 plus 1/2 is equal to 1. Instead of adding probabilities, you'll want to multiply probabilities. When the books are asking about the probability of multiple things happening, That's when you multiply the individual probabilities. For example, with our coin, our imaginary quarter, it's 1/2 for the first heads times 1/2 for the second heads would be 1 out of 4. Which makes sense. If you had 2 quarters, you could do heads both of the times. You could do heads and then tails. You could do tails and then heads. You could do 2 tails. Heads and heads would be 1 out of 4 possibilities. We're saying the same thing. It's just a matter of which way you want to do it. Now, if you were looking at the probability that we flip heads 3 times in a row, I am not listing out all 8 possibilities of what you can get with your 3 imaginary quarters. Suffice it to say 1/2 times 1/2 times 1/2 equal 1/8. You would be multiplying the probabilities. Now, returning to our previous genetic example, what's the probability that 3 offspring will each have the genotype of big T, big T? We're going to multiply our probabilities. Remember, the probability of being big T was 1 out of 4. Times the probability of being big T is 1 out of 4. Times the probability of being big T is another 1 out of 4. When you start multiplying everything out, we get 1 out of 64. This is where I need the calculator. I'm sorry, I haven't bothered with a calculator for years. We're just going to call it 1 out of 64 and go on with life. Then the lab book asks, what is the probability that 3 offspring will all be tall? Now, we had been working with the probability of the genotypes. We're now asking about the probability of a phenotype being tall. We should probably add your phenotypes into the boxes. So we've got 3 talls and 1 short. So the probability of being tall is going to be 3 talls out of 4 boxes. The probability of being short is 1 short out of 4 boxes. The probability that the lab book is asking for is 3 tall kids. The probability of the first kid being tall is 3 out of 4, times the probability of the second kid being 3 out of 4, is 3 out of 4. The probability of the third kid being tall is 3 out of 4. Multiplying it out, we get 27 out of 64. Multiplying it further, yes, I do know how to use a calculator, thank you very much, is 42%. If you've got 3 kids, odds are one of them's going to be short. 58%. No, Miss Naples. Look. The first kid is that first box is going to be tall. The second box is going to be tall. The third box is going to be tall. And I don't want a short kid because I'm not having the fourth one. It doesn't work that way. It's a probability. You might have the first kid be short, and then you just kind of ruined your whole experiment. Maybe the second kid is short. Maybe the third kid is short. Maybe the fourth kid is short. Maybe the 42nd kid is short. Oh, wait, we were only supposed to be doing 3 kids there. But odds are 58% of the time you're going to get a short kid in one of the first 3 kids. It just kind of works that way. Probability is a little weird. While the probability of each child being tall is 3/4 or 75%, the probability that 3 children being tall is only 42%. Odds are one of those first 3 kids will be short. It's not like you can just do away with that last box. In fact, when we start talking about a lot of the genetic diseases, people find out they're heterozygous, carrying a recessive allele, often because their first kid has the 2 recessives for some sort of genetic disease. That's how a lot of people find out their genetics, is they've got a kid who's homozygous recessive for something bad. Not that short's being bad, but yeah, sometimes you get that in other diseases. At any rate, number 5, monohybrid sex-linked inheritance. In humans, as well as other primates, monkeys, sex is determined by special sex chromosomes. An individual containing 2 X chromosomes would be a girl, while an individual containing an X and a Y chromosome would be a boy. What's your genotype? You're going to have to pick yours. I don't care. Pick one. Let's see. What gametes are produced by a female would be X, X. What gametes are produced by a male would be X, Y. Which parent determines the sex of the child? We can do a Punnett Square just like every other Punnett Square, although, yes, I kind of labeled the X's and the Y's in red and blue so you can tell who was who there. If we've got our red X's along the top and our blue XY along the side, you pull the red X's down, nothing changes. The female can only give an X chromosome. There's really nothing to distinguish the sex of the child. Ladies, all we can give is an X chromosome. On the other hand, when you add in the gametes from the male, on the first row, we've got the blue X's going across. If the guy donates a sperm with an X chromosome, he's going to get daughters. On the bottom row, we've got the blue Y chromosome. If the guy donates a sperm with a Y chromosome, he can have sons. It's actually the guy who determines the sex of the child. Us females might carry the baby in the uterus, but we do not determine the sex of the baby. That's on the guys. That's their responsibility. They can donate a sperm with an X chromosome and get daughters, or they can donate a Y chromosome and get sons. Not our fault, guys. Y'all did it, not us. Now, these sex chromosomes do bear alleles for traits just like the other chromosomes in us humans. But genes that occur on the sex chromosomes are said to be sex-linked. Usually we're talking about a gene on the X chromosome because in real life the X chromosome is a lot bigger than the Y chromosome. Trivial knowledge for trivial minds, the X chromosome contains about 1,500 genes, whereas the Y chromosome contains about 230-something genes, most of which are associated with determining male or female there. But the X chromosome's a lot bigger, so it has a lot more genes. So for our purposes in non-science majors class, we're going to say sex-linked genes are on the X chromosome. Blah, blah, blah. The Y chromosome is smaller than the X chromosome. Consequently, most of the genes on the X chromosome are absent on the Y. There's a couple of things that we can talk about. In humans, color vision is a sex-linked trait, and the gene for color vision is located on the X chromosome, but absent on the Y chromosome. When working with sex-linked traits, you do have to indicate the dominant big N allele and the recessive little n allele, the X chromosome and the Y chromosome. So it gets a little more complicated. An X with a big N is going to be a chromosome with normal color vision. An X with a little n is going to have a colorblind allele on it. The Y chromosome just indicates male. It does not have a big N or a little n on it. Just a Y chromosome. He's too small. He's missing that spot. So what we're going to do for letter A down here is normal color vision, X with a big N, is dominant over colorblind, X with a little n. Suppose a colorblind man has children with a woman whose genotype is X with a big N and X with a big N. Okay. We need to figure out the genotype of the colorblind man. Because he is male, he is going to need an X and a Y chromosome. Because he's colorblind, his X chromosome gets a little n. So his genotype would be X with a little n and Y. We know he's going to be crossed to a X with a big N, X with a big N woman. We just needed to know the genotype of the colorblind male. The box for the Punnett Square is actually on page 208. You might have to flip back and forth a little bit. This Punnett Square is a little more complicated. You do have to keep track of your X's and your Y's. You do have to keep track of your big N's and little n's. Try to keep them all together. So we'd have the gametes from the first parent say that X with a big N, X with a big N, normal color vision, homozygous normal color vision woman would go on the top, and the gametes from the colorblind male would go down the side, the X with the little n and the Y. We're still going to meet the gametes in the middle, and we're still going to fill in all of our boxes. Try to keep your X with a big N in front of your X with a little n, and whatever you do, make sure your big N is a little different from your little n so that you don't get too confused later. Once you've filled in the chart, then we need to know about the probabilities. The question is going to ask what proportion of the daughters would be colorblind. Note that first row does have two daughters who are heterozygous, but they've both got at least one big N, so they're both going to see normal color vision. So none of the daughters, 0 out of 2 of the daughters, would be colorblind. What proportion of the sons would be colorblind? At this point, kind of ignore the girls and look at that bottom row. We've got an X with a big N and a Y, an X with a big N and a Y. Hey, we've got some boys, we've got some big N's, we've got some normal color vision. The proportion of the sons that would be colorblind would be 0 out of 2. The lab book doesn't actually ask about all the kids, but what would the probability be that any of the kids would be colorblind? Well, there's no girls, there's no boys, so it would still be none. But in this case, it would be 0 out of 4. Rather than the 2, we'd be talking about all the kids, so if you do put the 0 down, make sure you put it 0 out of 4. And no, it's still 0 in math class, but it's 0 kids out of 4 boxes. Probability, see? On page 208, letter B, one of the daughters from the previous question marries a colorblind man. So, what are the genotypes that we're going to use on our Punnett Square? One of those F1 daughters. Remember she was X with a big N, X with a little n. Is going to be crossed to a colorblind man. He's still going to have an X and a Y, but he's going to have a little n. Just like that. It is not Dad. She's not marrying Dad. Yes, I've had that question asked before. She's marrying a guy with the same genotype. She is not marrying Dad. Thank you very much. Get your mind out of the gutter, people. Yeah. Anyway, you should fill in your Punnett Square on page 208. Hopefully, you got the same thing I did there. The next question is what proportion of their sons will be colorblind? Ignore the girls for just a moment. We'll look at that bottom row there. We've got one with X with a big N and a Y. Oh, that's normal vision. X with a little n and a Y. Ooh, colorblind. On the proportion of sons that will be colorblind, it's one colorblind son out of 2 sons would be 1/2. Letter C. Oh, not letter C. Explain how a colorblind daughter might result from the above marriage. How do you get a colorblind girl? Oh, hey, look at that upper box on the right-hand side with that colorblind girl. Where does she come from? You get an X with a little n from both parents. Same place you get all the kids from. One from each parent. Same thing. Why you don't see as many of the colorblind girls? Because we've got 2 X chromosomes and the other X can help cover for a recessive allele. It is possible to get girls who have a recessive sex-linked trait. It's a lot easier to get a boy. They're not exactly half and half, but half of the potential genotypes would give you a recessive genotype. On the girls, it's 1 out of 3 phenotypes that get you the recessive. There's some more complicated stuff like gene frequency and stuff like that we're not talking about, but it's a lot easier to get a colorblind recessive, sex-linked recessive gene in males than it is in females. Your lecture instructor may go into more detail on that. At the bottom on page 208, letter C, a woman who's heterozygous for the colorblind gene marries a man with normal vision. What's the genotype of the woman? Well, she's going to be a girl, so she's going to have 2 X chromosomes. They said she was heterozygous. Heterozygous means one of each, different. So we've got to have an X with a big N and an X with a little n. We're going to cross her to a normal male. They don't actually give us a blank for that, but somewhere you've got to figure out the genotype of the guy, too. He's going to be a boy, so he's going to have an X and a Y, but he does have normal vision, so it's X with a big N and a Y. The Punnett Square is on page 209. Feel free to fill that out real quick while I wait. Hopefully you got your Punnett Square all filled out. Assuming they have 4 kids, what proportion of the children will be colorblind? So what proportion of the kids? The first box, X with a big N, X with a big N, nope. X with a big N, X with a little n, nope. X with a big N and a Y, nope. Ooh, look at that lower right-hand box there, X with a little n and a Y. There's going to be 1 out of 1, 2, 3, 4 kids would be 1 out of 4. What proportion of just the sons will be colorblind? At this point, ignore that top row because we don't want to look at the girls. We're going to look at the boys. And we do have 1 out of 2 sons. 1/2 for the boys. What proportion of just the daughters will be colorblind? At this point, ignore the bottom row, and that first girl's fine. The second girl's a carrier, but she's fine. 0 out of 2 girls will be colorblind. Sometimes it makes a difference whether we're asking what proportion of the kids, what proportion of the sons, or what proportion of the daughters have a problem. Especially on the sex-linked questions, read through the questions very carefully. Make sure you're answering the correct one. See if we're asking about kids, sons, or daughters. On page 209 and 210, the lab book goes through some dihybrid problems. Ms. Grady does not ask dihybrid problems on the lab exam, so we're just going to omit that, skip on to the questions on page 211. If your lecture instructor does go through dihybrid crosses, Ms. Staples does watch your lecture stuff, you may have to check with your lecture instructor and they may talk about dihybrids, but you don't need to know it for lab. Although it ends with a 9 to 3 to 3 to 1 ratio. On page 211, question number 1 says that in peas, the seed coat can be smooth or wrinkled. A wrinkled seed plant is crossed to a smooth seed plant. The F1 generation is all wrinkled, so which allele is recessive? Smooth or wrinkled? Well, think about it for a minute. If you cross a dominant to a recessive, well, if you cross a homozygous dominant to a recessive, that'll always give you that dominant phenotype out. So if we're crossing wrinkled to smooth and we get all wrinkled, wrinkled would be the dominant. Of course, the question's asking about which is recessive, so you should actually circle smooth. Why did we leave a big old blank when you didn't even need a Punnett Square? I don't know, habit. Question number 2. A wrinkled seed is test cross. The F1 generation is all wrinkled. What's the genotype of the parental wrinkled seed plant? So, we need our code again. A big W is going to be wrinkled, and a little w is going to be smooth. Try really hard to make your big W's really big and your little w's really little so you can tell them apart. Wrinkled is going to be test crossed. Remember that if you're test crossing, you're crossing it to a recessive. Our wrinkled could be big W, or it could be big W, little w. Which of those two gives all wrinkled? You might have to work some Punnett squares out. You can work the 2 Punnett squares out and see which one you think it is. When you work the 2 crosses out, big W crossed to little w gives a whole bunch of heterozygous but wrinkled plants. If you cross the other one, where you cross the big W to the little w, you get some wrinkled and some smooth. So it was going to be that homozygous dominant that gives you all the wrinkled offspring. So the question was asking what's the genotype of the P1 parental plant? Is big W. The question also asks what's the genotype of the F1 offspring, would be big W, little w. On page 212, number 3, 2 wrinkled seed plants are crossed. The F1 generation has 80 wrinkled seed plants and 20 smooth seed plants. What's the phenotypic ratio of the F1 generation? And what'd you cross, and what are the genotypes? And yeah, this one's going to be a little bit more of a think about it. We've got 2 wrinkled seed plants that are crossed. It gives an 80 to 20 ratio. What cross was being made? Well, first of all, what ratio is 80 to 20 closest to? First, you should probably try reducing down to its lowest common denominator and dividing both sides by 20. So you'd end up with 4 wrinkled and 1 smooth. Wait a minute, that's like 5 boxes. How do you get 5 boxes? you don't get 5 boxes. And sometimes in real life, the plants do not give exactly precisely the right ratio. If you flip a coin 100 times, you should expect to get 50 heads and 50 tails, but it wouldn't surprise you too much if you got 49 heads and 51 tails. Yeah, that's where we're going with this. But our ratio is a 4-to-1 ratio. What's it closest to? All wrinkled plants? 3-to-1, 1-to-2-to-1, 9-to-3 to 3-to-1, 1-to-1, all smooth. It's closest to a 3-to-1 ratio. So, if you're crossing 2 wrinkled seeds and it gives a 3-to-1 ratio, what cross was being made? Hint, you're crossing 2 dominants and you get some dominant and some recessive. 3 dominant and 1 recessive. Which one did I say made that work? If you have to work them all out, you probably need to stop the video and work them all out. Hopefully you remembered that crossing 2 heterozygous individuals was the way that you cross the 2 dominants and get some dominant and some recessive. If you needed to work everything out, hey, that's fine too, but you should have come up with letter C, where you're crossing 2 heterozygous individuals, so big W, little w, cross to big W, little w. Question number 4. Right-handed is dominant to left-handed. If you cross 2 heterozygous individuals, what's the probability of getting a left-handed child? You might have to write your genotypes out. Right-handed is dominant, so we'll be using big R's for right-handed and little r's for left-handed. We're going to be crossing 2 heterozygous individuals. So what genotypes are we crossing? Big R, little r to big R, little r. So fill out your Punnett Square, and then we can talk about what's the probability of getting a left-handed child. Once you've filled out your chart, you can see that only the bottom right-hand box has the left-handed child, so the probability of being left-handed would be 1 out of 4 kids, 1/4, 25%. The question goes on to ask what's the probability of getting 2 right-handed children. The probability of having a right-handed child to start with is 3 out of 4, times the probability of having another right-handed child would be 3 out of 4, would be 9 out of 16. Question number 5 is a dihybrid cross. You do cross 2 parental generations. You do get a heterozygous individual. You do have to self-fertilize. You do get a big, huge 16 square Punnett Square. And ultimately, the probability of getting a plant that has yellow pods and smooth seeds is 1 out of 16. How'd I get that? Don't worry about it. Just omit that one. But they're asking about the last box on the right-hand side. On page 213 is an optional problem. If you are not liking genetics a bit, just omit it, close your book, it's fine. If you're liking genetics, it's a really cool problem. It looks like it might be a trihybrid. It's actually not. If you think about what genotypes you can actually get from each parent, it's actually just a little 4-square Punnett Square. Yeah, there's the answer. We're not really doing that one. No, it's not on the test or anything.